So, hello everyone, and welcome to the webinar on seaweeds and honeybee health. My name is Dara Scott. I'm the Managing Director of Advanced Science, the company that makes HiveAlive. In case you don't know, HiveAlive is a feed supplement for honeybees based on seaweed extracts. That's why we're talking about seaweeds today. Just to first tell you a bit about myself, I have a strong background in medical diagnostics and also in marine research, mainly out of the world-renowned Woods Hole Oceanographic Institute out of Cape Cod in Massachusetts. I've been a beekeeper for over 15 years, and I was first introduced to beekeeping in New Zealand. Manuka honey, you've probably heard of, is a big industry there. And there are hives everywhere. I hadn't seen that before. I'm from Ireland, and in Ireland, beekeeping isn't as popular. So when I was there, I thought it was a really cool symbiotic relationship between bees and man. We help the bees out, and the bees give us some honey. So when I came back, I connected up with a professional beekeeper, spent the summer with him, and by the end of the year, we caught a caste swarm, and I still have their descendants today. So, as I said, home for me is in the west of Ireland on the edge of the Atlantic Ocean, and it's probably the worst place in the world to keep bees, mainly because it's wet. It rains here all the time, and I really mean all the time. When they say it rains a lot in the northwest of the US, they have no idea what real rain is like. We have huge problems just getting queens out to get mated because it can rain for 3 or 4 weeks solid, and they can't get out and fly. And even when it's not raining, it's windy, and this is the summer I'm talking about. So it's tough going, but I don't care. I love working with bees. Every time I go out, if there's something interesting, I'm learning something new. It's very interesting. So I suppose it's the two interests of marine research and beekeeping that led me to HiveAlive, and I'll talk a bit about that later on. So I'd like to thank Veto-Pharma for hosting the webinar. We work closely with Veto-Pharma. There are distributors for HiveAlive in the US and in Canada. You probably know them through their key product, which is Apivar. It's a Varroa product. I think it's the most popular Varroa product worldwide. I'm sure you've come across it in the US or in Canada. This is our first time running a webinar, so please bear with us if there are any technical issues. And just so you know, I'm doing this from Ireland, so it's currently midnight, but that's fine. I'm a father of 2 small children. I don't get any sleep anyway, and I like talking about bees anyway, so that's fine. And finally, I'd like to remind you that the webinar is recorded, so you can listen to it again afterwards. I know I talk too fast. I'm going to try and talk as slowly as possible, and also there'll be a lot of information. It's not a long webinar. It'll be about 20 minutes or so. But there is a lot of information, and you might want to look back on it again afterwards. So thanks for joining us, and I hope we all learn something new. So to give me a second, and I'll get the slideshow set up, and we can do that. Right, so today I'm going to talk briefly about seaweeds, their benefits to humans, and animals, and of course bees. I'll then talk about how seaweeds can be used in bee health and discuss the benefits that are seen in field trials using HiveAlive. Just so you know, everyone attending the webinar this evening is automatically entered into a draw. The prize is a $100 beekeeping supplies gift certificate, and at the end I'll announce the winner, so good luck to everyone. And after that I'll go through any questions you might have. You can ask a question by typing them in on the chat option there on the right hand side of your screen, and I'll do my best to get through them all. If anything else pops up in your head tomorrow or when you're working the hives over the weekend, please feel free to email us, as we'd love to hear from you, as we're constantly learning from beekeepers and the beekeeping community, and your feedback is genuinely very important to us. So, why seaweeds? Seaweeds have been eaten by humans for at least 18,000 years. Pretty much anywhere that seaweeds are available, their use has been documented as food and as medicines. The long-lived Japanese are famous for eating seaweeds in foods like sushi. And as I mentioned, I'm from Ireland. Ireland has a long history of seaweed use. Been used for hundreds of years to help plants grow, especially relevant along the west coast of Ireland, where the quality of soil isn't as good. One species of seaweed is traditionally mixed with honey and lemon and used as a medicine for colds, flus, and bronchitis. I had thought this was only an Irish tradition, but recently discovered that this is also done in a number of countries, even as far away as Venezuela. Nearly every country that has access to seaweeds has used them for medicines. From Malaysia to the Caribbean, New Zealand to North America, Greece to Japan, they all have had a tradition of using seaweeds for medicinal use. This can be used for a range of conditions, from lung disease to gut parasites, and from tumours to even syphilis. Most recently, scientists discovered that individual seaweed extracts have been found to be beneficial in digestive health and in tackling diseases such as diabetes, osteoporosis, and cancers. So, seaweeds and animals. A lot of animals will eat seaweeds naturally, but up until recently, seaweeds in animal feeds wasn't that widespread, possibly due to the volumes needed to feed animals. Seaweeds have been fed to animals traditionally for their nutritional content, but it's only in recent times that science has become more advanced, and we know exactly what parts of seaweed are most beneficial for animals. Since the discovery, the demand for seaweed extracts has rapidly increased. Very interesting compounds are now extracted from seaweeds. For instance, seaweeds have large amounts of polysaccharides. These are complex sugar molecules that can do lots of amazing things, like protect the lining of the gut or help increase good probiotic bacteria found in the gut. A number of seaweeds have excellent antibacterial, antifungal, and antiviral properties. More recently discovered, seaweeds have been shown to have immune stimulatory properties, which help animals better defend against disease. All these benefits lead to better overall animal health, and a result of having better health, productivity is increased. Nowadays, seaweed extracts are tailored to the specific animal for specific properties. For instance, in fish farming, seaweed extracts have been fed to reduce the attachment of a parasitic lice. In beef farming, extracts are used to increase weight gain, improve gut health, nutrient absorption, reproductive health, and milk quality. The same is done, or similar is done for horses, sheep, pigs, poultry, and seaweeds are even now fed to pets like dogs and cats. So, it's obvious that seaweeds are beneficial to animals and humans, but what about honeybees? Oh, by the way, as an aside, since we developed HiveAlive, we've been told by many beekeepers that they have seen bees feeding off seaweeds, which we didn't know when we were developing HiveAlive. So the bees must think that seaweeds are good also. But anyway, is there any research to show the effects of feeding seaweeds to honeybees? Now, it turns out there is. Previous research had discovered that certain seaweed extracts were successful in blocking the entry of the malaria parasite into human cells. So recently, a group of researchers wondered, could this theory be applied to honeybees? They set up experiments to find out if feeding seaweed polysaccharides, the polysaccharide I talked about a minute ago, could block the attachment of Nosema cells that causes internal issues in bees. They found that by feeding different seaweed polysaccharides, Nosema was reduced and honeybee survival was improved. They believe the seaweeds are able to do this by blocking the adhesion of the Nosema spores to the gut wall, thus stopping their life cycle. They also said that the seaweeds may improve survival and decrease spore levels by activating the immune system response. We're popping up a link on the right-hand side there to the paper if you're interested in reading it lately. I think it calls on the heading of a French study, yeah. So this brings us nicely onto the research that Advanced Science has done on seaweeds in developing HiveAlive. It's important to say at this point that HiveAlive is not registered as a medicine. It's registered as an animal feed, and as a result, we're not allowed to make any reference to treating or preventing diseases such as Nosema or any other diseases for that matter. So anyway, as I mentioned, I have a history in both beekeeping and marine biology, so I wanted to know how could the benefits of seaweeds be used to make a product that could help bees. I was keen to make a product that was natural based, but coming from a science background, I wanted to ensure that it had the science to back it up. That has always been the fundamental ethos of what science does. It's natural products validated through science. Ireland, being an island, has a lot of seaweed, and as a result of that, has some of the top seaweed researchers in the world. I began working with some of the key researchers, and together we came up with a number of seaweed extracts that demonstrated promise for feeding to honeybees. I knew intestinal well-being and digestive health was an issue in most beekeeping operations. If that isn't right, the bee will never be healthy and strong, so that's the area I focused on primarily. There were a number of aspects in choosing the best seaweeds and how to blend them. First of all, we had to find them. We only ever use Irish seaweeds. The strains we finally selected were the most beneficial for bee health, and some of these are unique to Ireland. So the extraction process had to preserve the bioactive properties of the seaweeds, so we developed a cold process extraction to obtain the key extracts that we needed for HiveAlive. We then trialed the extracts in cage trials and collaborated with organic chemists to perfect the blending process. At the time, I hadn't heard about bees feeding off seaweeds, so we were worried that the seaweeds might not be attractive to honeybees. But it wasn't an issue. We found out that bees actually consumed more of the seaweed syrup than the control syrup. We named our blend of seaweed extracts Ocean Shield to highlight how the seaweeds were protecting the bees. This blend was carefully formulated with thyme oil and lemongrass oil. Thyme oil has been used in beekeeping for many years and has a number of benefits on its own. Lemongrass oil is added to increase the attractiveness of the syrup. We chose the concentration of lemongrass oil carefully. We've included enough to be attractive, but not too much, which may lead to robbing, which we heard can happen with other products. Our quality control is at the highest standard, and we are fully EU approved and FDA compliant, and all our facilities are regularly audited to ensure compliance. So our initial cage trials found that adding seaweed to the syrup improved bee intestinal well-being. We then moved on to short field trials in Ireland with professional beekeepers. Feedback was really positive, and we realized we were onto something. We then progressed to longer, independent, large-scale trials, which I'll talk about a little bit later on. So before I get into the trials, I suppose I should tell you a little bit about why one would use HiveAlive. First off, I don't think I've said already, it's a liquid feed that's easily added to sugar syrup. By using HiveAlive, your colonies become stronger, more productive, and giving you, in the end, more bees and more honey. It's proven to maintain intestinal well-being, which keeps bees healthy. It's shown to help colonies survive the winter. It even prevents syrup from fermenting. This means you don't have to worry about syrup going bad or going off, and in particular, don't have to worry about syrup going bad in the comb, which can cause problems for colonies, especially if winter comes on fast and the bees have a chance to dry up and cap off the syrup. HiveAlive can be sprayed or drenched. We find this particularly good in the early springtime, and we recommend doing this once a week for 3 weeks to cover a full breeding cycle. It can even be used to help introduce new queens. So the best time to use HiveAlive is in the fall. This is when the bees are storing the syrup for winter. Because the bees are eating over the winter, they're eating it for a longer period of time, and so are protected for a longer period of time also. We recommend feeding in the spring to boost colony production. We first introduced HiveAlive to beekeepers at the Apimondia Bee Health Symposium 5 years ago. And now, due to demand, it's sold in over 30 countries and stocked by the largest beekeeper retailers worldwide. For our US listeners, you can get it in Man Lake, Brushy Mountain Bee Farm, Better Bee, and Miller Bee Supply. And for our Canadian listeners, HiveAlive is available through Propolis, Dickey Bee Honey Supply, and Urban Bee Supplies. A full list of our international retailers can be found on our website, which is hivealivebees.com. So to date, over 3.6 million colonies worldwide have now been fed HiveAlive. It's very popular with all types of beekeepers, from the hobbyist who just wants to protect the bees, the commercial beekeeper who wants to increase productivity and minimize losses. In New Zealand, for example, there's a big commercial beekeeping industry for Manuka honey. Over 45% of the beekeepers there are using HiveAlive annually. You can find feedback from beekeepers who have used HiveAlive on our website, and if you have any feedback to offer, please do contact us. Feedback in the US has been really good, and particularly in Canada, where a lot of commercial beekeepers are now using it annually. We're convinced the main reason for our success is the strong evidence that our product actually works based on scientific trials we've carried out to support its use, and realistic scientific trials, which I'll go through now. So first I'll talk about our largest and longest trial. It was a trial run by Dr. Hadagina in the Hellenic Institute for Apiculture in Greece. She's one of the key researchers worldwide on pesticides, and she works with the European Union in assessing the effects of neonics on bees. The trial was a long-term trial, and it ran for 18 months. The idea was to see how well colonies did with and without HiveAlive. It was independent research, and this study was peer-reviewed and published in the foremost honeybee journal, the Journal of Apicultural Research. The results were presented at the American Beekeeping Federation in 2015 in Los Angeles. The full paper can be found on our website, and we're also popping up a link now directly to where the paper is published. It should be, again, on the right-hand side of your screen. So we'll go through the trial together. There were 40 colonies used in the trial, 20 control and 20 treated. The only difference being that the treated group had their syrup activated with HiveAlive, and the control group didn't have their syrup activated with HiveAlive. One of the most important points of this trial is the number of hives we used. It's far too easy to skew data when working with small number of hives due to the variability of colonies. So we need to have big numbers of colonies to get realistic data. And we want to keep our colony numbers high, which we did. Both the groups, the HiveAlive group and the control group, received 4 liters or 1 gallon of syrup in the fall, and the same 1 gallon of syrup in the spring. HiveAlive was added per instructions, which is always 2.5 mils per liter or 10 mils per gallon. During the trial, colony population measurements were taken, which is what's on the graph here. For all our trials, there are multiple time points to clearly illustrate if there is a consistent trend. Again, a lot of studies tend to cherry pick time points which suits the results they want, but we have consistent multiple time points. So in the graph here, the yellow or orange line, depending on the color it comes on your screen, is the HiveAlive fed population over time, and the blue line is the control population. You can see the colonies were equalized in the beginning, and by the end of summer, you can see there's already a steady increase in population of the HiveAlive group compared to the control group. After the second winter, they were fed in the November. They started feeding, and then it went through into the spring, summer, and then they were fed again November again, and through the winter, and then springtime they were fed again. But by the springtime of the second winter, the HiveAlive group had 89% more bees than the control group. That's nearly twice the population than control. This data suggests that HiveAlive isn't a quick fix, and best results are seen when HiveAlive is fed each year to strengthen colonies. Now, a lot of people say that, oh, I'm moving hives all the time, or I'm splitting them and I won't see or appreciate the benefit, so it's no good to me. But it's actually quite the opposite. It's under these times of stress that the bees get the most from HiveAlive, and the resulting colonies will be all the stronger for it. So in the same trial, let's talk about overwinter mortality. During this trial, 3 of the control hives did not survive the winter, whereas all of the HiveAlive colonies did. That's a 15% reduction in overwinter mortality with HiveAlive. If you take the time to look at the published trial data, you can also read more about how the intestinal well-being is maintained. So we knew from developing the product in Ireland that HiveAlive worked in wet climates, extreme of that, and we now knew it worked in hot, dry climates, but we wanted to try it out in climates less extremes. So we were delighted when Veto-Pharma, our distributor partners, asked to run a trial in France to evaluate our product before they became our distributors. So Veto-Pharma ran a trial in Chalac in France, in the middle of France. They have a test apiary there, and they set it up there. Their climate was considered temperate, like, say, for instance, Ohio or New York City. They carried out a 12-month trial. There were 18 in control and 17 treated hives in the trial. And again, the colonies were fed 1 gallon of syrup in the spring and 1 gallon of syrup in the fall. So let's go through the results again. So again, colony population was looked at, but in addition to colony population, we looked at brood coverage and honey production and the amount of wax drawn out. The first graph you're seeing here is showing colony population. This time the HiveAlive group is in red and the control group is in blue. The green bars show the percentage the population gained when adding HiveAlive to syrup. So it's the difference between the control group and the HiveAlive group. As you can see, the HiveAlive population gets increasingly larger over time when compared with the control. By the end of this trial, the HiveAlive group had 20% more bees than the control group. But this trial was only run for 1 year, as opposed to 1.5 years for the Greek trial. So to compare, if you look at a year into the Greek trial, we also saw a 20% increase in colony population. So the results are consistent. Now, moving on to the next parameter that was measured, that's the amount of frames with honey and pollen stores. Again, the HiveAlive group is the red line and the control group is the blue line. By the end of this, we can see the HiveAlive fed group had approximately 45% more honey or pollen stores. We're assuming this is most likely due to the increased number of workers in the hive. We also looked at the coverage of brood on the frames. Again, we can see here that the HiveAlive fed group was larger by nearly 40% than the control group. Finally, the ability of the colonies to draw out wax was also monitored in this trial. It was found that just 3 weeks after feeding HiveAlive, the HiveAlive group were drawing out 7% more comb than the control group. So this completes our major trials to date. We have a number of ongoing trials which we'll publish as soon as we have the results, but it takes time to do these right. We have lots more information on our website on these trials, and also lots of information on the other lab work we carried out on HiveAlive. So do please feel free to check this out. And then to summarize what we've talked about. So seaweeds are used the world over as foods and as medicines. They're rapidly gaining popularity as animal feeds due to their antifungal, antibacterial, and immune stimulatory properties. There's data to suggest that seaweeds can help control nosema. We've gone through how HiveAlive was developed, talked about the benefits of using HiveAlive and when to use it. And to summarize the data that we obtained, feeding HiveAlive in the fall and in the spring results in increased colony population, increased honey production, reduced overwinter mortality, increased brood production, healthy, strong colonies, and finally, improved intestinal well-being. And that's it. So thanks very much for attending the webinar. I hope you found it interesting. If you haven't done so already, can you please send in your questions on the chat and we'll do our best to answer them. And as a thank you for attending, we're offering, we have a winner for our $100 gift certificate. It's Steve. So Steve, whoever you are, we have your email address. We will be contacting you to find out where you want the Beekeeping Supplies gift voucher to go to. If we don't answer your questions or if you have more queries, you can contact us. HiveAliveBees.com is the website, or you can email info@advancedscience.com. And of course, you can keep up to date with us on Facebook and on Twitter. We're pretty active on that. So we'll try and get through some of your questions. Oh yeah, we also have a frequently asked questions page on our website, so there might be some answer for that too in case we don't answer all your questions. Okay, thank you. So the first question we have here is, how much HiveAlive should I feed? That's a really common question. We get that a lot. Ideally, we recommend that all syrup fed is activated with HiveAlive. But we realize that this can be, cost prohibitive to the beekeepers that are feeding huge amounts of syrup. So the minimum we recommend is, like our trials, is the 1 gallon of activated syrup, which is basically 10 mils of HiveAlive added to 1 gallon. And primarily, if you can feed it at all, feed it in the fall. That's the best time to feed it because, like I said, the bees will be eating it for a longer period of time. But you can also feed it in the spring as well. We also have protein patties. Do you recommend putting in protein patties? That's from that you feed the bees. Oh, sorry. Do you recommend putting in protein patties or fond that you feed the bees in wintertime? Yeah, we can do that. Like, for instance, one of our Canadian retailers, Dicky Bees, adds HiveAlive to the protein patties already. We're actually looking in Europe here to see if we can add it to fondant and supply a HiveAlive fondant as it is. We've done trials with that in the past and it's worked out well. If you're doing it to patties, we recommend around 5 mils of HiveAlive per pound of patty. We have another question here. The bottles seem expensive for a small amount. Okay. Well, basically, for a start, HiveAlive is quite concentrated. You only need 10 mils or 2 teaspoons per gallon syrup. So depending on the size of the bottle, it can work out about less than $1, depending where you buy it, less than $1 to feed HiveAlive. Just so you know, there's 3 product sizes. In the US, there's a 100 mil, which feeds 10 hives. There's a 500 mil, which feeds 50 hives. And there's a 2-liter bottle, which feeds 200 hives. We do produce a 10-liter bottle for a couple of countries, but that feeds 1,000 hives. We haven't brought it into the US yet, but we may produce these for the US if there is demand. What's the shelf life of HiveAlive? The shelf life, it's 2 years from date of manufacture. But once you open it, we recommend you use it within about 6 months. That's the best thing to do there. And syrup from preventing syrup from fermenting. How long? So if you're preventing syrup from fermenting, we regularly will make up syrup in the autumn. If we don't use it in the autumn, we'll use it again in the springtime. So a good couple of months there before you can need to use it up. Are your trials conducted by independent body? Yeah. So the Greek trial was conducted by independent researcher, like I mentioned, Dr. Hagina. The Hellenic Research Institute that was independent of us. And the second one was run by Veto-Pharma, and they were basically assessing us to see if they wanted to take on their product and see if it was good enough for them to take it on. So yeah, independent trials, they were done. And can strong concentrations be harmful? We have used higher concentrations, but the concentration that we have is the ideal one, and we'd recommend using the ideal. Sometimes we recommend using, it's on instructions as well, using a higher concentration for when you're drenching it. That's okay. Don't go over that concentration that we recommend for drenching it. But the ideal is the 2.5 mil per liter or 10 mil per gallon. I think that's all our questions, is it? Someone's looking for recipes to make their own protein patties. You know what? There are loads of recipes on the web to make protein patties or fondants. Everyone has a different version. Just the key thing, I suppose, I think we have it on our instructions as well, is the concentration of 5 mils per pound of patty. So can HiveAlive be fed for 1 to 2 months in the spring and also for 1 to 2 months in the fall, or is that overkill? Valerie, no. There's no, if you add the right concentration of 2.5 mils per liter or 10 mils per gallon, you cannot overdose them. There's no issue with that at all. You feed as much syrup as you like. There's no problem with that at all. Okay, I think that is it. I think we'd just like to thank everybody. I'd like to thank Joanna very much from Veto-Pharma, and Linda very much here with Advanced Science, who's helped us pull this webinar together. It's quite the task, much more daunting than I thought it was going to be. And I hope everyone got something out of this. Hope everyone's learned something. And thanks very much for listening, and I hope you have a great evening. Okay, take care.